Hello, my lovely anatomist and physiologist, Michele Glass here, and welcome to our final installment of Chapter 3. And our topic here, of course, we're still thinking about the cell, and what we're going to be talking about in this particular video is what we can call the cell cycle. And when we think about the cell cycle, it's important to pay attention to the fact that the cell is the smallest living unit of life. And so everything, remember, that we do on the grand scale, right, as an organism, is happening in our bodies at the cellular level. So I always like to say, just like you're born and you mature and you get a job and maybe you reproduce and you die, cells are doing the same. So cells are formed, they have a maturation process, cells have a job that they do, some cells will divide, and there's definitely a window of life for each cell. So there's going to be a point when the cell will die, right? And so in this particular picture of the cell life cycle, we have, a whole circle, right? And 90% of the circle is a phase called an interphase. And so interphase is, we can say interphase is when the cell is doing its job. This is the time when the cell is maturing. This is the time when the cell is making more organelles. This is the time when the cell is making proteins. This is the time when the cell is doing whatever it is that cell does. So this is a red blood cell. This is when that red blood cell is transporting oxygen to your tissues. If this is a phagocyte, an immune cell that locates pathogens and engulfs them, the cell is in interphase if it's doing that particular thing. If this is a cell in your bone maintaining your bony matrix, that would be interphase. So interphase is the majority of the cell cycle. And then there's this tiny little slice. There's a tiny little slice, 10%, that's mitosis. And mitosis, we'll see, is this time of cell division, of specifically division of the nucleus. So if we start out with a cell that's just formed and just coming out of mitosis, very often that cell will enter a part of interphase called G0. So interphase is going to consist of G0, G1, that's hard to see, S, and G2. And the G here stands for gap. The G here stands for gap. And it's kind of a funny story. Scientists were looking at cells under the microscope, and we didn't have really great equipment at that point, and we didn't know a whole lot about the cell, and it doesn't really look like the cell's doing much during interphase, and so it was called a gap. And then mitosis looks really spectacular under the microscope, and so all the focus and study went there, and then everything else was just considered to be like a gap. And so now we know better. We know that the cell's doing quite a lot of business in the interphase part of its life cycle, and that's the majority of its life cycle, but that is the origin, that is the story of the G. So G0 is really just, I'm going to just say job. Okay, so this is when the cell is living its life, doing its job, transporting oxygen, maintaining bone, phagocytizing pathogens, whatever it is that cell does for the body. And there are times when it gets a signal that, oh, maybe it needs to divide. So maybe there's some damage that's occurred, and so we need to repair. Maybe the cell is just getting larger and larger and larger, and it needs to split into two cells. Whatever the signal is, if a cell gets a signal to divide, if it's able to divide, it will go into what's called G1. And G1 is part of this prep for division. If it continues going through the cell cycle, it will get to what's called S. And S here stands for synthesis. And I've highlighted it because this is when we replicate the DNA. And I mentioned that already. I mentioned a little bit about DNA replication in the previous video. And we said that was happening during interphase. So now we know it's happening during the S phase of interphase. And so let's think too about, okay, this whole stage of interphase is when the DNA is in the chromatin, because that's that relaxed, accessible form of our DNA molecule. And if DNA replication goes really well, we get a signal to go into what's called G2. And G2 tells us, okay, we got it. We're ready. Let's do the rest of the prep. I'm going to call it final preps for division. And then if the cell gets a signal, it goes into mitosis. And so mitosis is just this tiny, tiny, tiny little slice of time, right? We said it's like 10% of the life cycle. And this is when we actually divide the nucleus. And we're going to spend a lot of time talking about mitosis because remember, scientists thought it was so interesting under the microscope. They got real pizzazzy. And so that's been really well studied. So we're going to do that as well. So let's remind ourselves a little bit of detail about DNA molecules. So we did have our last video on DNA, so we should feel pretty grounded. Remember that chromatin is the relaxed DNA molecule. And when we're talking about DNA, remember we have the term genome, which is all your genes. And then we have the term gene, which is like the functional section of DNA. I kind of talked about it as like the recipe for the protein. Terminology is usually like codes for a protein type of thing. And it's again, when the DNA is in chromatin, that we're going to get DNA replication. We just saw that in S phase of interphase. And it was also when the DNA was in its relaxed chromatin that we were able to do that process of transcription that we talked about in the earlier video. It's not until we are ready to divide that we get chromosomes. So chromosomes are like DNA packaged around proteins. And it's part of, it occurs, or the DNA condenses during prophase. So I'm going to say formed during prophase, which we haven't talked about yet, so we will in a few minutes. And we'll see this then during division. So I'm going to draw like, here's my suitcase because we've packed up that DNA and it's ready for travel, right? It's ready for travel. When we talk about human cells, we're going to see that we have 46 chromosomes for humans. "46 chromosomes for humans." And what that means is we received 23 chromosomes from the egg cell, and we received 23 chromosomes from the sperm cell. And that gives us our total count of 46 chromosomes. So what that means is you're inheriting a chromosome from mom, and you're inheriting a companion. You're inheriting a companion chromosome from dad. And we're going to call these companions homologous chromosomes. And homologous chromosomes are going to be having the same type of information, but not identical. Why is it not identical? Because one came from the mom and one came from the dad. And mom and dad should be different. And humans, mom and dad should be different from each other, hopefully not closely related. And so all of the information that is inherited from the egg should be coming from the whole maternal gene pool, and all the information coming from the sperm should be coming from the paternal gene pool. And so it should be in your body a combination of these two ancestral pools, right? And so your homologous chromosomes are all these pairs. You get a number 1 for mom, a number 2 from mom, a number 3 from mom, and so forth. But you also get a number 1 from dad, and a number 2 from dad, and a number 3 from dad, and so forth. And so that's what we mean by homologous pairs. So very often, instead of saying human cells have 46 chromosomes, we'll say 23 pairs. And the pairs then, again, are talking about these homologous chromosomes. And you could be real abbreviated and say homologs. Sometimes it's easier to say, right? Remember. I like to think about my homologs as pairs of shoes. So I know they're a matching set. I know which shoes to put on. And I also know which one goes to my left and which one goes to my right because they are shaped differently. So within a pair of shoes, you have a left shoe and you have a right shoe, right? Same sort of idea. Remember, during S phase of interphase, we actually replicate that DNA. We're preparing for division. So we replicate that DNA. And when we do that DNA replication during S phase of interphase, then now we have two number 1s that we inherited from mom. We have photocopy. And we have two number 1s that we inherited from dad. So we are still having homologous chromosomes, yeah? But now we have what's called a sister chromatid. And sister chromatids are identical copies. So I like to just use that sister word and think about twin sisters, identical twin sisters. And so this is coming up, this is showing up during that S phase of interphase. So you could go back to this diagram and we could make note of that. We could say, okay, during S phase of interphase, during DNA replication, this is where we get our sister chromatids. Okay. Okay. Okay, the other thing that I need to make sure that we have defined here is this little kind of dot that I drew in here. So when I drew my chromosome, I have a little dot. And when I drew my sister chromatids, I had copies that were actually attached to each other. So another feature of a sister chromatid is that the chromatids are attached and that little dot is called the centromere. So we can say they're attached at the centromere. So I'm looking at my drawing of homologous chromosomes. I have an orange chromosome and I have a green chromosome. And the chromosomes I've drawn just like a line with a little rounded sort of bead maybe in the middle. And that little rounded bead is the centromere. And the homologous chromosomes are not attached to each other in any way. When you draw your sister chromatids, you do show attachment. So now I'm going to have two orange lines that are going to be actually touching each other at that centromere. So I'm going to draw that little kind of bead sort of in the middle, and I'm going to show that connecting the two orange lines. And then I'm going to have two green lines to represent my other set of sister chromatids. And again, I'm going to make sure that they're showing a connection at that centromere. Okay. All right. We mentioned the little egg cell and sperm cell, and we said that the egg is contributing 23 chromosomes for humans and the sperm is contributing 23 chromosomes for humans. And so we have a term for these type of cells. We can call them gametes, that's our fancy name, or we can call them sex cells. And again, we have either an egg cell or we can call site. And they're going to have 23 chromosomes. And then we're going to have a sperm cell, or it can be called spermatozoan. And it's going to have 23 chromosomes. And sperm cells have a head and then a flagellated tail. They swim. And when you have just 23 chromosomes in your cell, we describe this by the term haploid. And we can use a lowercase n to represent the term haploid. So the term haploid means you have half of your total chromosomes. The term somatic cell, on the other hand, soma means body. So somatic cells are all cells of the body except for gametes. These are your red blood cells. These are your white blood cells. These are your bone cells. These are the keratinocytes of your integument. These are your liver cells. These are your stomach cells. These are all of your cells except for either your egg or sperm cells. And your body somatic cells are going to have 46 chromosomes. And where did you get 46 chromosomes? 23 from the egg plus 23 of the sperm gives you your 46 chromosomes. And the term that we use here is diploid, and we use the abbreviation 2N to represent, meaning it's 2 times the haploid number, and the diploid number is the full set of chromosomes. Now these diploid haploid numbers do vary by species, but in humans, our diploid number is 46, as we've said, and our haploid number is 23, as we've said. And again, those numbers are not universally true, but those numbers are true for humans. Okay. We are actually ready now to talk about mitosis. So that was kind of like the big exciting thing that we promised to talk about with this whole video. Now mitosis, remember, is just like 10% slice of the cell life cycle. This is the process of nuclear division, meaning division of the nucleus, if you'd rather list that out. And it's happening in 4 stages. And you can use this abbreviation of PMAT if you'd like to help yourself remember these stages. So PMAT as in prophase, metaphase, anaphase, and telophase. Now, I like to consider that there's 3 major events happening in prophase, and I'm not listing them in a particular order. We're going to get the nuclear envelope to disappear. We're going to get DNA to condense into chromosomes. And we're going to get what's called the spindle apparatus forming, and it attaches to centromeres. The centromeres are those structures, remember, that are connecting the sister chromatids to each other. So I like to consider 3 major events happening during prophase. In metaphase, we essentially get the spindle apparatus to move chromosomes to center of cell. And we call this line where they're going to actually line up, and we call that the metaphase plate. And we can remember that meta here is referring to middle. In anaphase, we're going to see this spindle apparatus contracts, pulls apart sister chromatids, moves sister chromatids opposite ends of the cell. And then in our final stage, telophase, I like to consider that this is like reverse of prophase. So I'm going to write it in the same order I did above. So nuclear envelope reforms. DNA is going to relax back into chromatin. And then the spindle apparatus is going to disappear. So let's see what this would look like. So I think what I'm going to do is draw a cell and then progressively make changes to that single drawing. Probably in your notes, you want to have a series. No, I'm gonna draw the series. I'll draw the series. Okay, so I'm gonna start by just drawing like my circle of my cell, and I'm gonna draw a nuclear envelope inside, so a small circle inside. And I'm gonna then take 2 different colors. So I'm gonna use orange and I'm gonna use green to make little squiggles to show this is my starting cell. So in this case, I have DNA in a relaxed stage. So it's chromatin, I have a nuclear envelope intact. And I'm gonna show actually my centrioles. This is a part of our cytoskeleton, and this is where the spindle apparatus will form. So I'm showing the centrioles kind of in the cytoplasm of the cell, kind of across from each other on either side of the nucleus. As we go into prophase, what we're going to see happening is that nuclear envelope. So I'm repeating my picture, but now I drew my nuclear envelope, but I'm going to actually, no, I'm going to show it like it's disintegrating. So I'm going to draw my nuclear envelope, and I'm going to get my eraser to show it's starting to really disappear. So I'm making these big erasures way bigger than what would be the nuclear pore. And then I'm going to go ahead and draw, I've already had DNA replication happening earlier, so I'm going to draw one homologous pair. I'm going to use orange to represent one parent, green to represent the other. And I'm going to show that I already have my sister chromatids, and they have been, they are attached to each other at the centromere. And then I'm going to show my centrioles again, but this time I'm going to start to show spindle fibers coming out from that. So the spindle fibers are kind of like long points pointed in towards that nuclear envelope. Now, ultimately, during my prophase here, I'm going to see the nuclear envelope completely disappears and those spindle fibers attach to my centromeres. I'm going to next draw my cell. I'm going to show down the center. I'm going to have my orange sister chromatids in a line at the center of the cell with my green sister chromatids. I'm going to draw my centrioles on either side, and I'm going to have spindle fibers attaching to those centromeres. And here is my metaphase. And from here, I'm going to see those spindle fibers are going to contract. So now I'm going to draw my centrioles and my spindle fibers. But what I see happening is my orange sister chromatids are being pulled apart from each other towards opposite ends of the cell. And my green sister chromatids are getting pulled apart towards opposite ends of the cell. So I'm going to end up on one side of the cell with a single orange and single green chromosome, and on the other side of this cell I'm going to end up with a single orange and single green chromosome. And so this is my anaphase. And then to finish up, I'm going to draw my telophase. Give myself a little more room. And what I'm going to see here is basically two new nuclei. I'm going to get an orange chromosome in each side, and I'm going to get a green chromosome in each side. Ultimately, those would be in the relaxed stage of chromatin, but I don't want to illustrate that here because I do want you to see that you're getting exactly half of the genetic material in each one of those nuclei. Of course, my spindle apparatus has disappeared, and this is my telophase. Okay, so I learned this from a YouTuber. I think it's great. So I'm going to hold my hands together in like a prayer position. And I'm going to start then by crossing my fingers together to represent my prophase. And so I'm letting my fingers kind of stick out in a way to show that my DNA has condensed to form chromosomes. I'm going to remind myself that the nuclear envelope will disappear and that the spindle apparatus will form during prophase. Next stage is metaphase, and that's when my chromosomes line up in the center of the cell. And so here I am going to bring my hands together in prayer position. And then I have anaphase, which is when I'm pulling away, I'm pulling apart those sister chromatids towards opposite ends. And so I'm going to bring my hands away from each other. And then I'm going to bring my hands together to form two fists, as in I'm creating two new nuclei. This would be our telophase. And so remember, this is like reversive prophase. I've reformed nuclear envelopes, DNA is relaxed back into the chromatin, and that spindle apparatus has disappeared. So I love this prophase, metaphase, anaphase, telophase kind of hand gestures to help me remember. I have just a little bit more information to pass along, and then we'll close out this video. So just bear with me another minute. All right, we've been talking long. I'll try to get this wrapped up. The last little bits of information here. This process of cytokinesis is actual cell division. So what we see happening in animal cells is basically the two cells will sort of begin to pinch in half. So I'm showing nucleus on one side, nucleus on the other side, and then they're starting to sort of pinch in the middle. We call this pinch the cleavage furrow, and ultimately it will continue to pinch, continue to pinch, continue to pinch until you have two full cells. Now, sometimes cells go through mitosis and not cytokinesis, and in that case, they are increasing their number of nuclei. And we do see some cells are multinucleated, and that's how that would happen. The other thing that we need to mention is that we have in the cell what are called checkpoints in the cell cycle. And, excuse me, we don't have to go into all the details of the cell cycle checkpoints. You'll talk about that if you take a biology class. But what I want us to think about here basically is this is like, you know, we have basically this is a red and a green light, right? And so the checkpoints are going to start out as stops. It's like when you go through a toll. If you've ever traveled a toll road, you have to stop and then you pay your coin and then you get the green light and you're able to drive on. So the checkpoint is just like this. It's going to stop you first, and then you have to get signals to proceed. And so these are signals that can be coming from outside of the cell or can be coming from the cell. And the key thing to pay attention to here is that cancer occurs when your cell stops, I'm going to say listening. The cell stops listening to these signals, or the cell just pushes its way through the checkpoint, and that happens too. So the cell can make up its own story. And so this cell basically is going through unregulated cell cycle, is going through unregulated mitosis, uncontrolled cell division. And this can happen because a factor can be inherited, or this can happen from environmental factors, including viruses, can all cause this out of control cell cycle. The last little bit to talk about are what are called stem cells. And these are cells that maintain the ability to do mitosis. When we drew that first cell cycle, we said some cells go into G0 where they just do their job, and sometimes they stay there and they never get the signal to go back and do G1, S, G2, and mitosis. Some cells just don't have that ability. A stem cell is a cell that maintains that mitotic ability. It's mitotically active. But this is really significant. And a stem cell is unspecialized, meaning, you know, really its job is to do mitosis. Its job is to do mitosis. And so we have three varieties of stem cells to talk about. We have what's called a totipotent stem cell, a pluripotent stem cell, and then a multipotent stem cell. And we're going from a least specialized cell to a more specialized cell. So a totipotent stem cell is can become any cell. So we'll see this in embryonic development. In a pluripotent, they can become any type of tissue. But these are not able to create a whole organism. So they can't support development of an organism. And then the multipotent, we'll see, can become different cells within a lineage. So not any cell in the body, but maybe any kind of skin cell type thing. And here we have oligopotent variety, and then we have unipotent variety. And the oligo can be one of a few types, whereas the unipotent can make more of same type. So anywhere that we see tissue growth and repair, you have to have some type of stem cell that's present. And places in the body that cannot be repaired are lacking in stem cells. Okay, whew, we did it. That was chapter 3. As always, take care of yourselves and each other.